Welcome to the deep dive. Today, we're jumping into something really fascinating, how genetics is changing the way we grow our food.
Yeah, specifically molecular markers and plant breeding. It's quite the revolution.
And for this deep dive, our main source material comes from the Statistical Genetics course notes and transcripts. These are from Professor Guilherme da Silva Pereira over at the Federal University of Viçosa's Department of Agronomy.
Right. So think of this as our guide.
Yeah.
Our goal here is really to unpack the core ideas from Professor Pereira's course.
Kind of give you a shortcut, right?
Exactly. A shortcut to understanding how we're using this molecular data, this DNA information, to improve the plants we rely on, make things faster, more precise.
Okay, let's get into it. So plant breeding, at its heart, it's all about genetics, right? The
Yeah.
course material starts right there with why genetics is key,
Mhm.
and it highlights these two almost opposing forces, heredity and variation. So heredity, that's the passing down of traits, yeah? The genetic code carrying all that info.
Yeah, like yield, disease resistance, all that stuff. And then variation is just, well, the differences you see between plants.
Right.
But the key thing for breeders is, if those differences are genetic, if they're in the DNA, they can potentially be passed on. So heredity lets you pass things down. Variation gives you the options to choose from.
So selection, like inbreeding, it acts on that variation.
Mm-hmm.
But it's heredity that makes sure the good stuff, the traits you want, actually get passed on. Breeding is basically guiding that heredity.
Skillfully guiding it, yeah.
Yeah.
So how do we actually look at the genes to do that guiding? That's where molecular biology comes in.
Okay. And the course mentions the central dogma,
Yeah.
DNA to RNA to protein.
Right. The basic flow of genetic information, it gets complicated, obviously. But for breeding, the big leap was figuring out how to link specific DNA variations directly to what we actually see in the plant, the phenotype.
Ah, okay. So finding that DNA signature for a good trait.
Exactly. If you find that signature, you don't have to wait for the plant to grow up, which could take, seasons, sometimes years.
Right.
You can just look at the DNA from a tiny seedling
Right.
and sort of predict its potential. It's revolutionary being able to read that blueprint.
And that leads us to things like QTLs, quantitative trait loci.
Yes.
Yeah.
QTLs. The course material explains them as basically regions of DNA that influence complex traits, things like yield or drought tolerance, stuff that isn't controlled by just one single gene.
Okay, so finding those regions is powerful, but how do breeders actually use that info?
Well, that's the next step, and it's a big one. Marker-assisted selection, or MAS.
MAS.
This is where we use molecular markers. These are like identifiable variations in the DNA sequence itself. We find markers that we know are physically close on the chromosome to, or linked with, a gene or a QTL that controls a trait we want.
So it's different from traditional breeding where you just look at the plant and cross your fingers, essentially.
Pretty much. The material gives a good example. Let's say you cross a disease-susceptible plant with a resistant one.
Okay.
If being susceptible is dominant, then in that first generation, the F1, all the plants might look susceptible. They carry the trait, but it's hidden.
But somehow the resistance gene's hidden away.
Exactly. With MAS, you use markers, or linked to that resistance gene. You test the DNA of the seedlings in the lab, maybe from the F2 generation.
Ah, so you can see which ones have the resistance marks.
Before you even plant them out, you
Yeah.
identify the promising ones super early.
Wow. Okay, that must save an incredible amount of time and resources. No need to plant thousands just to see which ones survive a disease.
Fundamentally changes the game. Saves time, space, money, everything. Now, to actually do this, you need the right tools. The course notes mention enzymes like polymerases to copy DNA.
Uh-huh. Nucleuses to cut it, ligases to stick it back together.
Right. And techniques like PCR, polymerase chain reaction, to make millions of copies of a tiny bit of DNA. Gel electrophoresis to sort the DNA fragments by size. And DNA sequencing, of course, to actually read the A's, T's, C's, and G's.
The molecular toolkit.
Yep. And with these tools, we find the markers themselves. These are just spots in the DNA where individuals differ. Older markers were things like RFLPs, RAPDs, SSRs. Might ring a bell for some listeners.
Okay.
But now, with cheaper, faster sequencing, next-generation sequencing, the big focus is on SNPs, single nucleotide polymorphisms.
Just single letter differences in the code.
Exactly. and you can find millions of them across the genome. Plus, they're super cheap and easy to detect now with things like genotyping by sequencing or GBS.
Okay, so let's walk through the course outlines. You make your crosses, create a population with mixed traits.
Right, your breeding population.
Then you collect data on both the phenotype, what the plants actually look like, their traits,
Ah.
and the genotype. So you use those molecular tools to see which markers each plant has.
Mm-hmm. Then you build a genetic map showing where the markers are on the chromosomes.
And then the statistical genetics part kicks in.
You got it. Using statistical methods to find links, associations between specific markers and the traits you measured. That's QTL mapping, finding those important DNA regions.
And once you have markers you trust that are reliably linked to good traits.
You use them. That's MAS. In the next breeding cycle, you select plants based on their marker profile from the lab tests, not just waiting to see how they perform in the field. It speeds everything up and makes it way more precise.
So connecting this back, thinking about how precisely we can now tailor plants, reading their genetic code like this, it really opens up some big questions, doesn't it? Like, what possibilities does this unlock for the future of food, of agriculture?
It really does. I mean, this material from Professor Pereira's course, it underscores how central molecular genetics has become. It's driving so much in modern ag and biotech.
So here's something to chew on, a final thought based on all this. As we get even better at reading and using this plant DNA information, how might that change the actual variety of crops we have available? Or maybe how resilient they are, especially with our climate changing so rapidly.
That's a really interesting point to consider. Diversity and resilience in a changing world.